Welcome to GNP Sir classes. In continuation to our previous class on Animal Nutrition Unit 2, Dr. Pramod Kumar explains the micronutrients in this class. Listen carefully and learn animal nutrition for your composite exams and JRF Animal Science. So the first element we discuss is sulfur. Sulfur, S, is a structural component of skin, hair, wool, feathers, cartilage and connective tissue. It is present in amino acid, cysteine, cysteine and methionine, hormones, oxytocin and insulin, vitamins, thiamine and biotin, glutathione and in the inorganic form in chondroitin sulfate of cartilage. The sodium-sulfur ratio in ruminant diets should be approximately 10:1 to 15:1. In the case of sheep, the ratio of N/S is 5:1. Wool is rich in cysteine and has 4% of sulfur. Deficiency symptoms: reduced feed intake and cellular digestion, protein malnutrition, poor quality of wool in sheep. Now we talk of elements that are called electrolytes and include sodium, potassium and chlorine. So first we talk of sodium. Sodium, Na+, is the main extracellular cation of the body. The sodium pump, Na pump, controls electrolyte balance and is a major part of the basal metabolic rate in the body. It also plays a role in the muscle contraction, transmission of nerve impulses and in the absorption of sugars and amino acids from the digestive tract. Sodium is included in the animal diets as sodium chloride. Deficiency symptoms: poor growth and reduced utilization of digested proteins and energy. Bone softening, keratinization of corneal epithelium, impotency in males, impaired estrous rhythm in females, cardiac output decreases. In laying hens, lowered egg production, weight loss and cannibalism can be seen. Sodium, potassium and chlorine combinations. All of these elements are involved in the maintenance of osmotic pressure and acid-base balance, regulate the passage of nutrients through cell walls and help in water metabolism in general. There is a regular dietary need, as because of limited storage, excess is being rapidly excreted. Deficiency symptoms: lack of appetite, a decline in growth, loss of weight and production in the adult. Chlorine. Chlorine is the major anion found in extracellular fluid. It also plays an important part in the gastric secretion, where it occurs as hydrochloric acid as well as chloride salts. Chloride is supplied through sodium chloride in the animal diet. It is excreted from the body in the urine and is also lost from the body, along with sodium and potassium, in perspiration. Deficiency symptom: a dietary deficiency of chlorine may lead to an abnormal increase of the alkali reserve of the blood, alkalosis, caused by an excess of bicarbonate, since inadequate levels of chlorine in the body are partly compensated by increase in bicarbonate. Reduced growth. This table shows the electrolytes sodium, chlorine, and potassium, their major effect, deficiency and toxicity. Potassium. Potassium, K, is the major intracellular cation. It plays an important part in nerve and muscle excitability and is also involved in carbohydrate metabolism. Na and K are interrelated in metabolism. K gets efflux from the cells whereas Na gets influx inside the cells. Deficiency symptoms: in chicks given experimental diets low in K, retarded growth, weakness and tetany, followed by death. Deficiency symptoms, including severe paralysis, have also been recorded for calves given synthetic milk diets low in K, general weakness. Now let's discuss the microminerals or trace minerals. Microminerals, also known as trace minerals, are essential minerals required by livestock in relatively small amounts, i.e. less than 100 parts per million for various physiological functions and metabolic processes. These minerals are crucial for the overall health, growth, and production of livestock animals. Copper. Sources: pasture. The copper content of pasture varies from 4 mg per kilogram to 8 mg per kilogram of dry matter. Other sources of copper for livestock are seeds and seed byproducts, straws, little, liver meal, meat meal and fish meal. The copper content of milk is low. Distribution: muscle contains half of the total body copper, also stored in the liver and bone marrow. Concentration in the blood is 0.1 mg per deciliters. Functions: component of hemocupre, maintenance of the integrity of RBC, ceruloplasmin, binding of iron to transferrin. Along with iron, copper is necessary for hemoglobin formation. Component of metalloenzymes cytochrome oxidase, synthesis of phospholipids, tyrosinase, melanin synthesis. Feather pigment taurusin. Essential for osteoblastic activity, collagen and elastin formation. The above picture shows the relationship of copper, molybdenum and sulfur. Metabolism of copper. The primary site for absorption of copper is enterocytes. It binds to serum albumin and amino acids transported throughout the body. Liver. Major storage organ, released from the liver for hepatic synthesis of ceruloplasmin, erythrocyprin and other enzymes. Diseases occurring due to copper deficiency. Copper deficiency leads to reduced iron absorption with resultant microcytic hypochromic anemia, pigs, chickens, microcytic nomochromic anemia, calves. Depigmentation of wool, hair and feathers, reduced tyrosinase activity, steely, stringy wool in merino sheep, lack normal crimp. Enzootic ataxia, sway back in lambs, kids and calves, degeneration of motor neurons. Copper deficiency also results in falling disease in cattle, chronic, leg-sooked, scouring disease, copper pine wasting disease of cattle, sheep. Dissecting aneurysm of aorta in chickens, lack elastin formation, aorta becomes weak, impaired reproduction, low fertility. Toxicity of copper excess. Pigs are highly tolerant. Cattle, goat less tolerant. Sheep are more susceptible than other animals, especially those fed palm kernel cake, develop toxicity. Iron. Sources of iron. Green feeds, grasses, leguminous fodder, meat meal, fish meal, blood meal, liver, egg yolk, milk and byproducts, colostrum, 3 to 5 times than milk. Distribution. Hemoglobin, 60-70%, myoglobin and enzymes, cytochrome C, peroxidase, catalase. Ferritin, 20%, and hemosiderin, 35%, are stored form of iron. In blood, serum is transferrin. Hemoglobin in cattle, poultry, pigs, 11 to 12 grams per deciliter. Sheep, goat, horse, 10 to 11 grams per deciliter. Egg, 1.1 milligrams iron. During fetal life, neonates utilize iron stored in the liver. Functions. Components of several enzymes: cytochrome, electron transport, xanthine oxidases, activation of oxygen, catalase and peroxidase, breakdown of peroxides. Functions of iron. Components of several enzymes: cytochrome, electron transport, xanthine oxidases, activation of oxygen, catalase and peroxidase, breakdown of peroxides. Transport of oxygen by hemoglobin. Iron supplements can detoxify gossypol. 1 gram of hemoglobin carries 1.34 milliliters of oxygen. Metabolism of iron. Iron is absorbed from the duodenum by two-stage process: mucosal and serosal transfer. Mucosal stage, hemoglobin is taken by the heme carrier protein, HCP1, and its iron is released by heme oxygenase into the intracellular pool. Non-heme iron in a ferrous state is reduced in the apical membrane and taken by the divalent metal transporter. Serosal transfer, accompanied by hephiston, copper-dependent homologue of ceruloplasmin, and a transport protein for aposhum, in the basolateral membrane. Theories for absorption. Mucosal block theory: the intestinal mucosa absorbs Fe during periods of need and rejects it when stores are adequate. B. Modified theory: the major regulator of Fe absorption is the Fe concentration in the epithelial cells of the duodenal mucosa. Deficiency diseases. Pigs, chickens: hypochromic microcytic anemia. Calves: microcytic nomochromic anemia. Piglet anemia 2 to 4 weeks after birth, kept on concrete stalls. So milk equals 2 to 4 grams Hb per deciliter. Piglet requirement equals 15 GHb per deciliter. Symptoms: pale mucous membrane, anemia, labored and spasmodic breathing, thumbs, mink cotton fur. Toxicity. Siderosis: excess deposition of the storage form of iron in the tissues. This is a chronic occupational disorder, for example persons involved in welding or iron mining. Hypersideremia, abnormally high plasma iron concentration, it damages to intestinal mucosal cells. Cobalt. The sources of cobalt for livestock are green feeds, grasses, leguminous fodder, meat meal, fish meal, blood meal and liver. Distribution. Of total cobalt, 43% is stored in muscles, 14% in bone. Cyanocobalamin and vitamin B12, 4.4% cobalt. Dietary essential in ruminants, synthesis of vitamin B12, functions. Synthesis of propionate by the ruminal microorganisms. The absorption, transport and cellular utilization of Vit. B12 is facilitated by CBL transporting protein haptocorrin, intrinsic factor and transcobalamin 2. This picture shows the metabolism of cobalt. Cobalamins leave the rumen as dead microbes or bound to salivary haptocorrin. They are released in subsequent digestion and selectively bound to IF in the abomasum. Adecobal acids methylmalonyl-CoA mutase to form succinate from propionate, thus facilitating gluconeogenesis chiefly in the liver. Deficiency diseases. Wasting disease cattle. Cobalt pine sheep, herbage less than 0.1 mg cobalt per kilogram dry matter, bush sickness, salt sickness, naturists, Lexhut, Grant Travers disease wasting condition. Normocytic normochromic anemia cattle, sheep. Toxicity. Toxic level in cattle 1 mg cobalt per kilogram weight. Sheep 3.5 mg cobalt per kilogram weight. Iodine. Source. The richest source is marine origin, seaweed, sea fish, 6 grams iodine per kilogram dry matter. Cereal grains, plants, leaf greater than stem, iodized salt, potassium iodide and calcium iodate. Distribution 80% iodine found in the thyroid gland. The iodine content of the thyroid gland on a dry matter basis is 0.1%. T4 forms 65% iodine. Functions. Synthesis of T3-triiodothyronine. T4, thyroxine, tetraiodothyronine. T3 is more potent than T4, T3 binds to nuclear receptors to a much greater extent than T4. The metabolism of iodine is shown in the chart. Deficiency diseases. Goiter, enlargement of thyroid gland, big neck. Goitre gens brassica family, L5, vinyl 2-oxazolidine 2-thione, signs calves, piglets hairless with thick pulpy skin. Birds molting and feather depigmentation. Infertility problems, males decline in libido, females decreased conception rate, suppressed estrus. Toxicity. Most animals appear to start showing ill effects when fed rations containing 200 to 500 ppm iodine. Manganese. Source. Roughages, cereal grains and byproducts. Manganese salts, oxide, chloride, carbonate. Distribution. Amount in the animal body is extremely small, found in bones, liver, kidney, pancreas and pituitary gland. Constituent of enzymes arginase, pyruvate carboxylase and superoxide dismutase. Functions. Synthesis of chondroitin sulfate. Activator of enzymes hydrolysases, kinases, thiaminase. Through activation of glycosyl transferase, is required for the synthesis of mucopolysaccharide that forms the organic matrix of bone. Deficiency diseases largely confined to poultry. Decreased chondrogenesis, bone abnormality. Swine crooked legs and enlarged hocks in Mn deficient diet. Porosis, slip tendon poultry chicks, Achilles tendon slips, chondrodystrophy parrot beak due to shortening of lower mandible. Toxicity. Considered least toxic trace mineral to birds and mammals. Mn concentration above 70 ppm is poisonous to animals. Zinc. Source. Animal protein, yeast, bran and germs of cereal grains. Supplementation. Zinc oxide, zinc carbonate, zinc sulfate, zinc methionine, organic source. Distribution. Present in epidermal tissues, hair, wool, milk and insulin. Constituent of enzyme carbonic anhydrase, 0.3% zinc, retinol reductase, alcohol dehydrogenase, alkaline phosphatase. Functions. Plays an important role in keratinization and calcification. Deficiency diseases. Pigs parakeratosis, incomplete maturation of keratocytes resulting in abnormal retention of nuclei in the stratum corneum. Cattle hyperkeratosis, thickening of the stratum corneum with an abnormal quantity of keratin. Chicks swollen hawk syndrome. Toxicity. Higher zinc in the diet reduces feed intake and induces copper deficiency. Sheep 1000 mg per kilogram diet. Pig 4000 mg per kilogram diet. Selenium. Source. Selenium yeast, wheat, alfalfa, forages, meat and fish byproducts supplemented as sodium selenite. Distribution. Constituent of enzyme glutathione peroxidase. Functions. Glutathione peroxidase catalyses the removal of H2O2, thereby protecting cell membranes from oxidative damage. GPX contains 4Se atom and forms a second line of defense after Wt. E acts as an antioxidant. Membrane-bound September iodothyronine 5C deiodinase type 1, ID1, transforms T4 to T3. Deficiency diseases. Ill thrift in lambs, loss of weight fed on Se deficient fodder. Nutritional myopathy, dystrophy of muscles. Exudative diathesis in chicks, gizzard myopathy in turkey. Stiff lamb disease, white muscle disease, Mulberry heart disease, heart necrosis in calves and lambs, muscle degeneration, weak leg muscle, a condition manifested in standing and staggering gait, stiff lamb disease, impaired reproductive performance. Toxicity. Se is the most toxic among trace minerals. Toxicity arises when the dietary level exceeds 10 to 20 parts per million. Plants Astragalus racemosus 14 g per kilogram dry matter. Neptunia amplexicollis greater than 4 grams per kilogram dry matter. Blind staggers acute. Alkali disease chronic, hoof malformation, loss of hair, emaciation. Degnala disease chronic dry gangrene on the face, tail and hooves. Molybdenum. Source liver, kidney, meat meal, grains, legumes, pasture. Distribution. Constituent of enzymes xanthine oxidase, aldehyde oxidase and sulfite oxidase. Absorbed from intestine and is excreted in urine and bile. Deficiency diseases. Not common due to lower requirement. Femoral head necrosis in chickens. Toxicity. Tetanus, peat scour in cattle, liquid feces with gas bubbles. Chromium. Source. Supplemented as chromium acetate. Insoluble chromium oxide is used as a marker in the digestibility trial. Functions. Known as glucose tolerant factor, potentiates the action of insulin and thereby functions in regulating carbohydrate metabolism. Decreases cholesterol and increases HDL. Chromium decreases blood cortisol levels during stress. Deficiency disease. CR deficiency may lead to decreased sensitivity of peripheral tissues to insulin, associated with diabetic-like symptoms. Toxicity. Tanning industry effluent contains high chromium, 2000 to 5000 parts per million, gets deposited in soil and water and causes toxicity. In animals, chronic chromium toxicity symptoms include skin contact dermatitis, irritation of respiratory passage, ulceration and perforation of nasal septum and lung cancer. Acute chromium toxicity, inflammation and congestion of the stomach, ulceration of the rumen and abomasum. Cats can tolerate 1000 mg per day of chromium. Hexavalent chromium is more toxic than the trivalent form. Fluorine. Source pasture herbage, cereal and grains. Distribution. It occurs in an apatite form as an integral part of the structure of bone and teeth to the extent of 0.04 to 0.06% in the adult. Fluorine gives rigidity to bones and teeth. Deficiency disease. Dental caries in humans, demineralization. Toxicity. Fluorosis. If drinking water contains greater than 1 ppm fluorine F gets deposited in bones and teeth, 10 times, bones and teeth turns yellow. Bone outgrowths from surface exostasis of jaw bones and long bones. Mottling of teeth if drinking water contains 2 to 5 ppm fluorine. Silicon. Next to oxygen, silicon is the most abundant element on Earth in quartz. Crystalline silica is the most abundant mineral in the Earth's crust. Sources. Cereal grains contain a lower concentration of C than leaves and stems of the same species. Functions. C is essential for normal calcification of chick bone. It plays a role in mucopolysaccharide synthesis and may function as a biological cross-linking, contributing to the structural integrity of connective tissue. Deficiency symptoms. Depressed growth. Deficient chicks show small cones. Atherosclerosis, osteoarthritis in humans. High levels of silicon in the diet of farm animals may be detrimental but not toxic. High levels of silicon in mature plants decrease digestibility and it is in the form of opal phytolates. Ruminants, it may develop siliceous renal calculi. Tin. Trace amount of SN is widely distributed in tissues and feeds. A deficiency of SN, under practical conditions with grazing ruminants, does not seem likely. Function. SN contributes to the tertiary structure of proteins or nucleic acids. Arsenic. Source. Inorganic arsenic incorporated into pesticide. Eating recently sprayed forage with arsenic-based herbicides. Another common source is when cattle lick the ashes of burned fence posts. Inorganic arsenic compounds such as arsenite or arsenate can contaminate groundwater and cause arsenic poisoning. Toxicity level of 5 to 10 mg per kilogram body weight is toxic. Cattle, sheep, horse, intense abdominal pain, diarrhea, or bloody or mucoid diarrhea, a staggering gait, and irregular or thready, weak pulse, dehydration. Vanadium. Vanadium is believed to be essential for the chick and rat. It is present in human enamel and dentin. Sources: whole grains, seafood, meat, and dairy products. Functions: regulation of Na plus K plus ATP As, phosphoryl transfer enzymes, adenylate cyclase and protein kinase. EDTA appears to act as an antidote to vanadium toxicity, possibly by preventing absorption from the intestinal tract. Levels of 25 to 30 parts per million in the diet are toxic to most animals, while the dietary requirement is less than 500 parts per billion. Nickel. Source: soil, nickel-containing fertilizer. Distribution: it is present in RNA in rather high concentrations. Average concentration in the earth crusts is 80 parts per million. Functions: Ni is essential for the urease activity of rumen microbes, catalyzes the hydrolysis of urea to yield ammonia and carbamate. Deficiency symptoms: chicks fed on a diet containing less than 14 parts per million nickel shows deficiency symptoms. Impaired liver metabolism, i.e. reduced ability to oxidize alpha-glycerophosphate, and unstructured degeneration of the liver cells, dermatitis and change in pigmentation of shank skin. Goats, if less than 100 parts per million in the ration, can cause a higher abortion rate, reduced growth. So this completes the Unit 2. Hope you enjoyed the lecture. Thanks for watching. Kindly share, like, and subscribe GNP Sir official YouTube channel. See you in the next lecture on Unit 3. Bye.